Welcome back to Environmental Science 546, Drinking Water and Human Health. Today is lecture number 16. We're going to continue our discussion on microbial contaminants. Last time I introduced bacteria to you. We'll continue to talk about bacteria, and specifically we're going to spend our time on E. coli today. If you take a look at our outline, here's where we are. Drinking water and infectious diseases, bacterial infections. We talked about cholera or Vibrio last lecture. Today we're going to be talking primarily about coliform bacteria, point number 2. So coliform bacteria, we introduced the term coliform bacteria early in the course. Coliform bacteria is more specifically known as bacteria of the genus and species Escherichia coli, or it's often referred to simply as E. coli. These are not bad bacteria. We use E. coli as an indicator of contamination, but by themselves, almost all E. coli are non-harmful to humans. These bacteria are bacteria that normally live in the digestive tract of humans and other animals, so they should be present. These bacteria live in harmony with the host, so they live in harmony with us in our digestive tract and are important because they aid in digestion. They break down food. They're important in the fermentation of food. Within our systems, where they're supposed to be in our digestive tract, E. coli are rarely a toxic problem. Instead, we know E. coli because they're an indicator of fecal contamination. They are present in digestive tracts of animals, so if we find E. coli in water, that means that our water is contaminated with fecal wastes of other humans or animals. So we use them basically as an indicator. Now, fecal materials do contain other pathogens. They're just very hard to measure. E. coli is very easy to measure, so we use it as an indicator. So that's the first take-home point of this lesson. Coliform bacteria are an indicator of fecal contamination. E. coli is not going to kill you. It's simply our indicator or our way of telling if we may have pathogens in our drinking water. So basically what happens when our drinking water sources become contaminated with fecal material, we run a test and E. coli will be detected. If our drinking water is not contaminated with fecal bacteria or with pathogens, we should not be able to detect E. coli. So we run a routine test. All public water systems do it. The Center for Disease Control does it, a very common laboratory test. If you take introductory microbiology in college, you do an E. coli test. So we do a total coliform bacteria count of water. As it rises, we know we have more contamination. To protect our drinking water from pathogenic bacteria, EPA has a coliform rule. This rule mandates public water systems to monitor for Escherichia coli. It mandates periodic inspections of water delivery systems for bacterial contamination, and we would primarily be doing an E. coli test. If the water samples that are taken by the public water system contains E. coli, the public water system has to do additional testing for pathogens, and they have to take action to make sure that E. coli is not present. Oftentimes that means chlorinate the water to kill all potential pathogens in the water. So we basically have a coliform bacteria MCL. Remember MCL, EPA standard? Our E. coli MCL goal is 0. We want no coliform bacteria present in water samples. We have no coliform bacteria present in drinking water samples. We're pretty much assured that our drinking water is not contaminated with human or animal waste. Our MCL, the way we do a test, our MCL is we use what's called an MPN test. Not more than 5% of the samples per month can be positive. Depending on how big your public water system is, that determines how often you take and do an E. coli test. But the EPA says that not more than 5% of the samples taken by your public water system per month can be positive. Now, as far as doing an E. coli test, you have a handout that I provided you to talk about the test for E. coli. See that handout. We generally use what's called multiple tube fermentation techniques, or an MPN technique, to determine E. coli, or we can use a membrane filter technique, abbreviated as MF. But we have some very common tests to determine the presence or absence of Escherichia coli in drinking water. So what's important here for E. coli is to remember that E. coli is really not a pathogen. It's an indicator. There are hundreds of different strains of E. coli. Now, if we just take a look at E. coli here, E stands for Escherichia. That's the genus. Coli is the species. And within a species, there are hundreds of strains. So there are hundreds and hundreds of strains of E. coli out there. 99.9% of the strains are harmless. They will not have a negative impact on humans. I need to bring this up because we have had some cases where there have been some strains of E. coli that are toxic to humans. And they are relatively new things that have been discovered in the last 25, 30 years. But for most of for our purpose in this course, E. coli is an indicator that we might have pathogens in our drinking water. And it's a test that is routinely done by the 74,000 public water systems spread across the country. Now remember, 99.9% of the E. coli strains are harmless, but we see an occasional mutated strain that might become a public health concern, and I'll give you an example of one right now. Several years ago, E. coli strain 0157H7 was found, and it was found that it gives off an exotoxin, and this exotoxin is harmful to humans. It results in bloody diarrhea. In severe cases, it can result in kidney failure, and it could possibly kill somebody. So occasionally we have a strain of E. coli that is bad, but that's not the general rule. Remember, 99.9% of E. coli strains are innocuous. They're not going to harm us. They're an indicator. On that 0.1%, if we go back to strain 0157H7, this strain was diagnosed in the early 90s, and in 1993, this strain infected 700 people in 4 states, and it was a major public health problem. 51 people developed temporary kidney failure, and 4 people died. And this particular strain was not traced to water. It was traced to uncooked meat. And you can go back and look at the internet and pull up this strain and talk about uncooked meat, and you could find out, a lot of information about the food chain that had this problem, and what the Center for Disease Control found out about this strain. But again, this is not normal. E. coli is really our friend. It really helps tell us if we've got problems with pathogens in our drinking water. A little bit later in 1996, 2,741 cases were reported, and 2,741 cases were reported. That resulted in 102 cases of kidney failure. We saw these things occur again in 1998, 1999, 2003, 2004, 2007, 2010, 2011, 2013. So this particular strain keeps popping up. Currently, we estimate that this E. coli strain probably impacts about 20,000 people per year in the United States and may cause about 250 deaths. And this strain traditionally has been linked to meat, uncooked meat. But recent research has shown that there are other sources besides meat that may harbor this particular strain of E. coli. Things like unpasteurized milk, unpasteurized apple cider, and if you go onto the internet and you do a little bit of searching under these two topics, you will find cases of this situation in the last 15 years. And there is some evidence that this strain also might be present in some drinking water. However, normal drinking water treatment, we use chlorine, that would kill this strain. So if we take a look at the water implicated, that's what I think is important. We had an implication back in 1989 in Missouri. And here we had a cold winter, we had frozen pipes, water leaked out of the pipes, and sewage contaminated the drinking water. And 240 people were infected with this E. coli particular strain. It resulted in 32 people hospitalized, and eventually 4 people died from this strain. We go forward a little bit to the next recorded incident that's tied to water. In 1991 in Portland, Oregon, people swimming in a lake, ingesting water, swallowing fecally contaminated water. This study suggested that numbers of bacteria needed for infection in people was very, very small. In this case, in 1991, 59 people were infected. And you see these incidents every other year since 1991. So there are instances where this particular strain, 0157 of E. coli, gets into our water supply. The one thing that I think is scary is water has been implicated with this one strain of E. coli a couple times, but the one thing that I think is scary is this last one. Kids with diapers, fecal material, kids going into water with diapers. That's why we chlorinate swimming pools and things like that. To kill fecal material and pathogens, we recommend chlorine in swimming pools. We recommend somewhere between 1.5 and 3 parts per million chlorine in pools to kill the pathogens generated by fecal material generated by kids with diapers. And again, you want to kill the pathogens in the fecal material that might be accidentally introduced to those pools. And you can simply do it with chlorine. Also recommend that you adjust the pH of the water to a pH of 7.2 to 7.6, and you will solve the problem. The one thing that really scares me is not the swimming pools, But if you take a look at the data in the last 15 years, if somebody's going to get sick from pathogens that were passed through fecal material, I think the most dangerous place for you to go is a water park, because you've got people, little kids in diapers, and this fecal material gets in the water in water parks, and then everybody else accidentally ingests a little bit of the water. And we see a lot of the cases of people getting sick because of water parks today. Okay, this tells you a little bit about E. coli. The take-home message is that E. coli is an indicator organism. It will tell us if we have fecal material present, if our drinking water has been contaminated with raw sewage, either from human or animal waste. The last part of this lecture was just anecdotal, but I think that the important part of the lecture is that E. coli is an indicator. It's a routine test that the 74,000 public water supply systems in the United States have to do very often. They have to do this. If they're a big public water system, they're doing it every few hours. If they're a small water system, they're doing it every few days. Okay, we'll continue talking about bacterial contamination of drinking water in our next lecture. Thank you for your attention.